Hello to my friends joining us via recording. Today we are talking about the first part of lesson number 10, which is all about the way that neurons work. Friendly reminder that when we are working on lesson number 10, this assignment has two parts, both parts due by Sunday. And lesson number 10, along with lesson number 9 and lesson number 11, that's what makes up our Unit 3 exam. I know we don't like to think about exams, right? But coming up fast is our next unit exam. So keep that in mind. By the end of today, we're about halfway through the material that we'll see on that next exam. To get us ready to talk about neurons, we are tying our lecture material into what we talked about in lab class related to neurons. So let's go through. I'm going to work my way down the list from top to bottom. And we're going to figure out which parts of a neuron each of these statements are describing. So let's start here at the top. When we're talking about the part of a neuron that receives messages, what was the name of the part of a neuron that receives messages? Get my pencil here. Yeah, absolutely. My receiving part of a neuron is those dendrites. And as we get ready to talk more about receiving messages, remember that messages that neurons receive are in the form of these chemicals called neurotransmitters. So here's a teaser for us looking ahead. Whenever we talk about the dendrites receiving messages, the dendrites are receiving chemicals. So the kind of channels, stop scribbling on for me. I'm going to clear it off and we'll start again. Whenever we're talking about the kinds of channels that we see on the dendrites, remember that those channels are receiving chemicals or receiving neurotransmitters. So on my neuron, or excuse me, on the dendrites of my neuron, we're going to gated channels that open up because my neuron received a chemical message. So far, we've said that the dendrites are the part of my neuron that receive a message, that message being a neurotransmitter. Hey, what about the other end of the spectrum, if you will? What if it's not about receiving the messages? What if it's about releasing the messages? Which part of my neuron spits out those neurotransmitters, those messages? Yeah, Kira's exactly right. The part of my neuron that spits out messages is called the axon terminal, or sometimes we saw it in the lab packet, it was called the synaptic knob, synaptic end bulb, anything that has this beginning part synaptic in it, that's referring to a synapse, where neurons talk to each other. So in a bit, when we start drawing some pictures of neurons, you will see one neuron's axon terminal, where it's spitting out neurotransmitters, right next to another neuron's dendrites, where it's receiving neurotransmitters. Yeah, absolutely right. Audrey mentioned the thing you have in your guided lesson and on the homework assignment, where we can have synapse number 1 and synapse number 2. So when we talk about the neurons that talk to each other, somebody's releasing neurons or neurotransmitters at their axon terminal. Somebody else has their dendrites there to receive them. Once a neuron receives those messages, it does a process called integration, where I like to say it mixes them all in the blender and it sees what spits out. If what spits out is a message that we should share, we need to send that message away from the cell body. Remember, the cell body is where the dendrites are attached to. So if I decide, because I do integration, or because I throw everything together in a blender, and I decide that I should talk to my neighbor, I'm going to send a message down my axon to tell my neighbor what I just heard. So my axon takes messages away, my dendrite receives messages, and at the very end of my neuron, I spit out messages that I'm sharing with somebody else at a place called the axon terminal. Now, to help my messages go more quickly down my axon, because sometimes those axons are really long, I make a myelin sheath. This is a fatty substance that covers my axon. to help me send a message faster. When I talk about my myelin sheath, what was the name of the kind of cells that made that myelin sheath? We've only got one option that are cells, right? Yeah, exactly. It's those Schwann cells, right? The Schwann cells. A quick lab reminder, because this might matter for your lab exam. Remember that we also have a kind of cell called oligodendrocytes. Oligodendrocytes do the same thing, but when we're in the central nervous system. So if you're in the brain or the spinal cord, what I use to make my myelin sheath is a type of cells called oligodendrocytes. If you're in the nerves like you have in your fingers, if we're in a nerve like we have in the arms, the kind of cell that's going to make the myelin sheath there is just Schwann cell. We have a lot of more Schwann cells in the body than those oligodendrocytes. I like that. Audrey mentioned the food service delivery called Schwann's. My grandparents were a big fan of food delivery from Schwann, so the Schwann's man would come and bring them food. That's perfect. All right, so by process of elimination, we know that the place where I don't have myelin, the very few places where I don't have myelin, these are called nodes of Ranvier. And tomorrow we're going to talk about this more. When a neuron is sending its message, that message is actually going to jump in between each of these nodes, each of these open places. So this allows messages to go much more quickly. Schwann cells cover up most of the axon except for these small places called the nodes of Ranvier. Now, it's one thing to put everything in words and to describe what the parts of the neuron are. It's another thing entirely to find them on a cartoon. And we're even going to draw our own cartoons later. So you'll get really good at drawing neurons by the end of our lesson here. So let's start. I'm going to circle some parts and I'm going to ask you all to help me label them. All right, here's the first part that I just circled here. Got the letter C going on. Which part of a neuron did I circle here in black? What will we call this part of the neuron? Yeah, exactly. This is the part of the neuron called the dendrites. The dendrites, remember, were the part that receive messages. So dendrites receive messages that another neuron is sending. I didn't specifically ask you to label this part, but this part in here would be my cell body, or technical term for it that you see sometimes used as soma. So there's the cell body of my neuron. Now, again, a part that I didn't necessarily ask you to label, I'm putting a big box around it. I've got a triangle-shaped space right here. It's not on your list of things that you had to label on this picture, but does anyone remember from lab what that particular part is called? Yeah, exactly. Several of us remember because it's really important. This is a part of the neuron called the Axon Hillock. So when we draw our own sketches of neurons, we will definitely draw a little triangle here. The Axon Hillock is really important because this is going to be the place where a neuron adds together all of the messages that the dendrites are receiving. So the dendrites receive all kinds of information. That information gets mixed up together here at the Axon Hillock. And this is where a neuron decides whether or not it should send a message down. So my long part here that's attached directly to the Axon Hillock, this whole long thing is the axon, right? So the axon is the long part where messages go away. When we get to the ends of the axon, so each of these little things we see here, these are my axon terminals. So I'll put a circle around these as well. These are the axon terminals, or the synaptic bulbs, or something having to do with the synapse. That's what we would call the structures down here. We also need to label the Schwann cells, and we need to label the nodes of Ranvier. I know this is going to get crazy here, but I want you all to grab a pencil and I want you to color or to try to circle the nodes of Ranvier. Can we find our nodes of Ranvier on this neuron? Yeah, I like it. I'm seeing some purple ones, seeing some blue ones, got some red ones. Awesome. Yeah, so those nodes of Ranvier absolutely are these little spaces that we filled in here. So the nodes of Ranvier, we can't have them without having our Schwann cells next to them. So we're going to talk about tomorrow probably how some of our neurons don't actually have a myelin sheath around their axon. For those neurons, they don't have nodes of Ranvier because every part of their axon has no myelin on it. If we have areas though with myelin sheaths, in this case made by our Schwann cells, then they always have these little spaces that we found here called those nodes of Ranvier. Nodes of Ranvier are going to help us out with something that we call the action potential. The action potential. So when I'm sending a message, remember potential means charge and action means we're moving. When this neuron decides it should talk to its neighbor, what we'll actually do is a kind of conduction where it's called jumping conduction basically, where the only places along this membrane that we actually move stuff in or out of the axon is only at these places that we colored. So the nodes of Ranvier are going to be really important when we talk about the way that neurons send messages inside themselves. When a neuron is sending messages, it's changing its membrane charge. Here's a word that we used before with muscle cells, and we're going to use it again here when we talk about neurons. Neurons are what we call excitable cells, which means they can get excited, which doesn't mean the same thing for a neuron as it does for my 4-year-old. But when we talk about an excitable cell, it means their membrane charge changes. An excitable cell is one that their membrane charge changes. That's how they get stuff done. When I'm talking about the normal membrane charge for a neuron, when it's not freaking out, what's the name of the normal charge? Or you can give me the abbreviation. We've sometimes used an abbreviation for it. What's that abbreviation or the name of the normal charge on a neuron? Normal charge. Yeah, exactly. So we've got the number and we've got the name. So the normal charge on a neuron, -70 millivolts. This is what we call its resting membrane potential. Resting membrane potential. So when a neuron is not receiving a message, when it's not talking to its neighbor and sending a message, a neuron's going to be at its resting membrane potential. So on our graph of the membrane, how the membrane changes its charge, when we see here at the beginning of the graph, where we're down here at -70, this is the part of my graph that represents resting membrane potential. Resting membrane potential. Sometimes I'll abbreviate it RMP. I don't make you learn a lot of numbers for this class. You do need to know this number. Must know this number. So if you haven't already, on your graph for membrane charge, underline, highlight, star, -70. We must know that. By the way, if I'm not talking about neurons, what if I'm talking about muscles? Muscles have a different resting membrane potential. Yeah, exactly. Several of us chiming in. When we're talking about muscles, so here's another underline, highlight, star for us. When we are talking about muscles, they're also excitable. They also change their membrane charge. But remember that muscles start a little bit more negative than neurons. So 2 numbers we have to remember for resting membrane potential. For a muscle, -90. For a neuron, -70. Resting membrane potential, when we are resting or waiting for another message. We start to receive some messages. These messages are really exciting. I reach a certain point where there's so much excitement, I realize that I've just got to talk to my neighbor. This negative 55 point on my graph, what do I call this point on the graph? Negative 55. That's another number we have to know, by the way, negative 55. Yeah, exactly. Negative 55 is really, really important. This is what we call the threshold value for a neuron. Remember, way back from unit 1, that if my membrane charge gets all the way to negative 55, we are now far enough away from normal that my voltage-gated channels are going to open up. Those ones that if the charge is wrong, they freak out, right? That's voltage-gated. Threshold is where voltage-gated sodium channels open up. And when they open up, all the sodium rushes inside till we get all the way up here to the top of my graph. This is the charge my membrane has to get to if I'm going to talk to my neighbor. I have to get all the way up here to positive 30. To go from negative 55 all the way up to positive 30, so when I'm on this slope of my graph, what do I call this process of getting more positive on the left side of my graph here? We're getting more positive. Yeah, Kira mentioned it for us. Temipeo is mentioning it too. Depolarization. That's when my membrane charge becomes more positive. We're moving away from what we started at. We're moving away from the resting membrane potential. That's depolarization. Once we get completely depolarized, though, we have this moment where we say, oh, I am not supposed to be positive 30. This is way too high. We will work to get back down to normal. Over on the right side of the graph, we're doing what's called repolarization. Repolarization, the goal being to get back to normal, to get back to the bottom. So this side of the graph is repolarization. We are so excited to get back to normal that we go a little bit too negative. When we go a little bit too negative down here, just like we say a solution can be too concentrated or more concentrated, we'd say that's hypertonic. Down here, where we become more negative than normal, is when we would say that we are hyperpolarized. So hyperpolarization here at the bottom of the graph, where it's just more negative than it's supposed to be. It has to do with the fact that the channels that bring me back to negative, they aren't good at closing fast. They stay open a little too long. We get a little too negative down here, hyperpolarization. We will talk about this graph more in our lesson today and tomorrow. But before we move on from it, are there any questions we can think of right now? Or give me a thumbs up if we feel okay about it. Perfect. Lots of thumbs up. Okay. Well, we will be back to this graph. And I know at least one of my groups was talking about how on the homework, this graph has letters at lots of different places to give you practice naming these parts. So you'll definitely do this activity again when you're working on the homework. As we prepare to talk about the way that neurons use ions to do their excitable stuff, to change the membrane charge, we always have to come back to the fact that our cells are like salty bananas. Now, I have to give kudos to one of my groups. They did a great job turning this into a banana. Here, I'm gonna follow their lead here, and we're gonna turn this cell into a banana. I liked how they even gave it the little black stem at the top. So kudos to my group who turned this cell into a really awesome banana. I was impressed. This banana is a salty banana. Where do you all want to start? On the inside or the outside? Where do we want to start? Kira votes outside. I'm cool with that. Ooh, Edith votes inside. Who's going to be my deciding vote? Everyone's like, don't pick me. All right, Carissa decided. Oh, outvoted. Okay, we're going to do it inside because I got a couple last-minute votes. When we talk about a salty banana cell, what do we find inside that cell? Sodium, potassium, or chloride? Who do I got a lot of on the inside? Yeah, exactly. Lots of potassium on the inside. And it's good that several of you put K+. This is going to be a chapter where it's really important that we remember the charges on everybody. So lots of potassium on the inside with a positive charge. Who do we have on the outside then? Lots of potassium inside. Who's on the outside? Yep, so I got that Cl-, and I've got that Na+. Na+ is my sodium. Cl- is my chloride. Okay, first thing that we must know is where stuff's found, right? Sodium and chloride on the outside, potassium on the inside. Now we need to figure out which way things are moving. When we talk about channel proteins do passive transport. Passive transport. What do we remember about passive transport? Passive transport with my channel proteins. There's a lot of stuff that I hope we remember with passive transport. Let's see what we remember. Okay, so Kira's mentioned a couple of ideas for us about passive transport. This was the kind of transport that didn't require any energy. Remember that active transport is where we use energy for. Passive transport, no energy required. When we do something that there's no energy required for, we're always going from high concentration to low concentration. So we're always going from where there's a lot to where there's a little. That's why we started with looking at where we had a lot of these things, because they're always going to move away from where we have a lot of them. So we said there was a lot of potassium that's inside the cell. Inside has a high concentration, which means if I open up a channel protein for it and it's doing passive transport, is potassium going to go out or going to go in? Since it's passive transport and we're going from high concentration to low. Yeah, a couple of us are voting. We're voting correctly. If I open up a channel protein for potassium, it's going to leave. I've got a high concentration inside, not as high of a concentration outside. Because of that, potassium is going to leave. So what does that mean about chloride? What's chloride going to do? Yep, several of us mentioning correctly. Chloride's coming in, going from where there's a lot outside to where there's a little inside. What's sodium going to do? My last one here on the list. What's sodium going to do? Yeah, sodium is also going to come inside too. Excuse me. When I bring sodium with its positive charge inside, we're doing addition. We're adding to the charge on the inside. So when sodium moves, when I open up a channel for sodium, that's going to make my membrane charge more positive. It becomes more positive. Because sodium has a positive charge, it brings that positive charge inside. Hey, chloride is also coming inside, but chloride has a negative charge. What was the name, by the way? Let's see if we remember, of things that have a negative charge. What kind of ion had a negative charge? Yeah, exactly. Several of us are remembering. The word we use to describe chloride is an anion. It's something with a negative charge. So when something with a negative charge, an anion, comes inside, it's going to make the inside more negative as well. So chloride comes in and we become more negative on the inside. We're coming in. The other thing that can make us more negative on the inside, though, is what potassium does. So potassium, like Temiteo mentioned, potassium is a cation. Potassium has a positive charge. It's a cation. But when potassium leaves the cell, it's kind of like we're doing subtraction. We're taking positives and we're putting them outside. So another way for us to make our membrane charge more negative is to get rid of some of those positives that we had inside the cell. So when potassium moves out, it steals its positive charge with it, making the inside more negative. When chloride comes in and brings its negative charge with it, obviously that makes it more negative as well. Sodium, it's positive, and that positive makes the inside more positive as well. This is going to be important when we start talking about, here's some ideas that we'll pull together, things called inhibitory post-synaptic potentials, or the way that honestly would be easiest for you to memorize is just IPSPs. These are changes in the membrane charge. Remember, that's what potential means. Post-synaptic means in my cell that's listening. Sometimes when a neuron sends a message at its axon terminal that opens up a channel for potassium, or that opens up a channel for chloride, we change our membrane charge in a way that makes us more negative. If my membrane charge becomes more negative because you sent me a message that made me get more negative on the inside, I would call my change an IPSP. So any neurotransmitters that open up channels for potassium or chloride, they're going to generate an IPSP, an inhibitory postsynaptic potential. We also have lots of neurotransmitters, though, that generate what are called excitatory postsynaptic potentials. Excitatory postsynaptic potentials, and those are abbreviated EPSPs. I got a little excited with my S's. I apologize. EPSPs, meaning my listening cell, the postsynaptic cell, changes its membrane charge in a way that's excitatory, in a way that makes it more positive. So if I'm a neurotransmitter and I attach to a channel that lets sodium come inside, that's going to generate an EPSP. My neuron gets excited. If I'm a neurotransmitter and I open up a channel for potassium, I'm going to generate an IPSP. My neuron's going to get sad. It's going to get more negative. Same deal if I attach to a channel for chloride. If I go and open that up, that's going to make the inside more negative. I'm generating an IPSP. Here's my wink, wink, nudge, nudge. Remember, when you take the exam, show me your blank paper, the front and the back, and then draw yourself a salty banana cell. You can even draw your little arrows, put your little positives and negatives. Go for it. Use this mnemonic as a way for you to remember, what do I have inside? What do I have outside? How's it going to move? And what's that going to do to my membrane charge? Thumbs up, thumbs down. Mind blown. Where are we at? Okay, getting some thumbs up. Hey, I was super excited to find this. Yeah, we got salty banana emojis. Pretty much made my day. Salty banana emojis. Day of class before we all take the final exam, right? We'll have to send each other salty banana emojis. Oops, I cut out there. I was just saying we'll have to send each other the salty banana emojis on that last day of class. All right, so I've talked about some of the stuff kind of from the middle of the packet, this EPSP, IPSP stuff. Let's actually take a step back. So we're going to go back to the beginning of your packet, some of the stuff that you learned about at the very start when you were working on the guided lesson. One of the first things that you learned about in the guided lesson was something we actually talked about in lab already, called the functional classification of neurons. Functional classification of neurons. When we talk about classifying neurons based on their function, we have three types. We have what are called the sensory neurons, their job being to collect as much information as possible, all kinds of information, sensory neurons. Sensory neurons don't they don't process the information they receive at all. They don't know what it is that they're collecting. They just collect it. That information is sent to our next kind of neurons, the neurons that do the process of integration. When we're talking about a neuron that does integration, we call that kind of neuron an interneuron, an interneuron. Interneurons are really good at, like we mentioned, this word integration, or the easy person word for integration is processing. So they put together all the sensory information that you collect, and they use it to predict what we should do about that. In this case, your eyes are collecting a bunch of information that there's a cup of water in front of you on the table. Your eyes see this. They don't know what it is. They don't know what to do about it. They send that information up to the brain, where my interneurons collect it and process it and figure out, I should pick up that cup of water. The neurons that actually make that happen are the motor neurons. So motor neurons don't decide what to do, but motor neurons make it happen, whatever the brain said we should do. Three kinds of functional classifications of neurons. The sensory neurons, all they do is collect information or perceive things. Interneurons that process it or predict how to respond or integrate that information. And then our motor neurons that do things like start muscle movement or do things like control, regulate muscle contraction. That's the kind of thing that motor neurons do. This is where I remind you, when you're working on the homework assignment and when you're taking the exam, to read questions really slowly, because I promise I will give you key words to help you figure things out. So again, like I mentioned with a sensory neuron, their emphasis being things like perceiving or collecting. When we talk about an interneuron, the big one is going to be processing or predicting. And again, with motor neurons, it's controlling, it's causing an action to happen, making stuff actually happen inside the body. That's the job of motor neurons. Yeah, it's all about action. So watch those words, read those statements closely. That will help you to figure out which kind of neuron we're talking about. In your body, you need all three of these types of neurons to work together for you to be able to function correctly. If they're not all working together, we can have a condition like congenital analgesia. Hey, help me out in the chat. How many of us had a chance to work on this lesson before class today? How many of us had a chance to watch the video about congenital analgesia? That's kind of where I'm going with this. Yeah, so several of us at least did a little bit on it. Yeah, so there's a video in the guided lesson that shows us what it's like if we have an issue with some of our neurons. So there's this medical condition that a video highlights a little girl with, her name's Gabby. She has what's called congenital analgesia. So this is a medical condition where a person doesn't feel pain. They don't respond to pain like normal. Pain, we're not really fans of, right? We don't like to be in pain, but the goal of pain is to keep you safe. So for example, you start stepping on something that's sharp, you feel the pain of stepping on it before you step all the way on it. It's going to cause you to move your foot away. Or you put your hand on the counter while you're playing on your phone, right? You put your hand on the counter, but you realize, oh, that's not actually the counter, that's the stovetop. You realize it's hot. You pull your hand away. Pain is supposed to protect you. We can have issues with multiple neurons in our functional circuit to make us not respond to pain correctly. So there's two forms of this I don't feel pain syndrome. The first form is called congenital pain insensitivity. Congenital pain insensitivity. When we talk about this form of the not feeling pain, what's happening in congenital pain insensitivity is that a person is not perceiving their painful stimulus. So they're touching a hot burner, but they're not perceiving it. Which kind of neuron did we say did perceiving? Which of my functional classification perceived things? Yeah, exactly. When we talk about perceiving something, that's the job of my sensory neurons. Sensory neurons collect information from your environment. If you're not collecting or perceiving the fact that something you're touching is hurting you, then you have what's called congenital pain insensitivity. First version of not feeling pain. You don't perceive the pain. You don't even feel it. Literally, you don't feel it. Second form of congenital analgesia is called congenital pain indifference. Congenital pain indifference. When we talk about congenital pain indifference, we do perceive that painful stimulus, but we don't respond to it. Now, before you type anything in the chat, don't type anything in for me. I'm going to set up an anonymous poll really fast. I would like you to tell me which kind of neuron isn't working. I'm going to give you a poll that's going to pop up for you. So your options are going to be interneurons, motor neurons, or sensory neurons. Let me deploy my poll. Completely anonymous, and if you don't know the answer, just give it your best guess. I don't care. In the poll for me, let's do a vote. Do we think that congenital pain indifference is an issue with interneurons, motor neurons, or sensory neurons? I don't care how you vote. I just want to see where we're at as a class. Most of us has voted. Promise no judgment. Poll closing in 3, 2, 1. Somebody's holding out on me. That's okay. See where we're at? Can you all see what we voted? Or is it just me? I'm hoping you all can see it too. Okay, you can see it too. Okay, so lots of us voted for interneurons being an issue. Lots of us voted for interneurons. When we voted for interneurons, what was some of the reasoning that we gave for that? Why could this be an issue with interneurons? Okay, so Audrey's saying we got a processing issue going on. Okay, can a classmate expand on that? An issue with processing? That is what my interneurons do. They do that processing. Okay, so yeah, we can feel it. We're choosing not to respond. Okay, yeah, so the idea that we know that it's pain, yeah, because we have in our text here, we know that it's painful, but we're choosing not to respond toward it. We're not predicting the right response. Yeah, I like that. So interneurons could definitely be an issue with congenital pain indifference. Check this out though. I have one brave friend who I'm slightly putting on the spot, but I'm going to enlist your classmates as well. This could also be an issue with motor neurons. Even if you didn't vote for motor neurons, how could you see this being an issue with motor neurons? How could this be a motor problem too? Maybe it's a processing problem, but maybe it's not. What if it's a problem with motor? Okay, yeah, so I like what Kaylin's mentioning. She's saying that the wrong response is happening. Maybe we know that it's painful. We know that it hurts, that it's hurting us. We know that we should do something, but we're not doing it. Maybe it's because the motor neuron's not working, right? The interneuron says, here's what you're supposed to do. And the motor neuron's like, nah, it's my day off. I'm gonna take a pass. So it could be that we know what we're supposed to do. We've predicted the right response and we just don't do it. If that was the case, that'd be an issue with our motor neurons. Now, if we were doing this in class, so it's a teenager neuron, Kaylin says, yeah, that could be. Nah, not today. I don't feel like doing what I'm supposed to. If we were in person, I 100% suspect that at least one of you would be like, okay, Dr. Ollis, so what's the right answer? Is it A or B? And I bet at least several of you are thinking that and questioning that right now, right? Is it A or is it B? This is an example of where it could be either. Yeah, so the correct answer for this one could be A or B. Now, before we freak out too much, I promise I would not put this kind of question on the exam where there's more than one right answer. I will not put this question on the exam. Yeah, so Kaylin gave a great rewrite. If I were to put this on the exam, the way that I would write it is I would ask you which kind of neuron is not malfunctioning, right? The only thing we don't know whether it's a processing issue or whether it's a motor issue, but we do know at least that it's not a sensory issue, right? So again, I will not put this kind of question on the exam. This kind of question is for us to think about and to do critical thinking together. So remember what your three types of neurons do. When we talk about perceiving and collecting, that's a sensory neuron thing. When we talk about processing or predicting, that's an interneuron thing. When we talk about triggering a response or causing a muscle to do something, that's going to be a motor thing. So this is good practice for you. I know you all hate my tricky questions, right? But this is good practice for you to look for those context clues. What does this question tell me? What do I know about these neurons to help me predict the answer? Yeah, exactly. So Kaylin mentioned the kind of information that would help us to answer this question is knowing what each of these things do, right? What each of our types of neurons do. So if I tell you they're not doing it, we can figure out who's slacking, who's not doing their job. Absolutely. I found this comic funny. I don't think I put it in the guided lesson. I'll give you a moment to read it, and then we'll talk about it. So it's important that the cells in your body all do their own thing. If muscle cells tried to be interneurons, it probably wouldn't work. Remind me in the chat, what is the normal job of interneurons? What do interneurons normally do? Any takers? What do interneurons do? Yeah, I like it. Krista talks about how interneurons are about processing. They take that sensory information and they predict what we should do. They process it. So imagine that instead of having interneurons, we got muscles. And the muscle cells, one of them says, sir, we're getting a signal from our neighboring neuron. What should we do? Any message that we get from our neuron neighbor friend, a muscle cell that's trying to process it is always going to say punch it. Because that's what muscles do, right? They just punch. They just contract. So it's good for us that we can have more than one type of response. Interneurons help us to figure out what we should do when we're not busy punching our other cells. That wouldn't be so kind. So interneuron funny for you there. When we talk about neurons, neurons always talk to each other. Neurons never live alone. They're always going to be in circuits. So to help us out with learning these circuits, we're going to do some terminology here. Pre-synaptic and post-synaptic neurons. Before we start drawing, friendly reminder for us that a synapse is a place where two neurons meet, or where they talk to each other, a synapse. So I've got one neuron that's called the pre-synaptic neuron. That's my neuron that brings a message to the synapse. And then I've got my post-synaptic neuron, which is the neuron that gets the message at the synapse. So if you haven't already in your notes packet, let's do some drawing together. And I'm drawing on a computer screen. So if I can do this, I know you can do this too. Here we go. We're going to start with neuron number 1, drawing in a nice little cell body. Remember, I'm going to put a triangle on it for that axon hillock. Here comes my big long axon, and I'm going to put those axon terminals on it. We should not forget our little friends, the dendrites, right? Here's my little dendrites. This is my presynaptic neuron. He's the one that's going to bring the message to the synapse. Now the synapse is only half drawn right here. I've only got half of my synapse because I just have axon terminals. I just have axon terminals over here. So I need to add neuron number 2. I need to add my postsynaptic neuron. So at the synapse, you have all the dendrites of neuron number 2. But each of my neurons then extends to an axon hillock and an axon and those axon terminals. This is my postsynaptic neuron. So big ideas when we are talking about a synapse. I guess I should label that, shouldn't I, here. This area is my synapse. When we're talking about a synapse, this is where the axon terminals of one neuron meet the dendrites of another neuron. If you are bringing the message, so if your axon terminals are at the synapse, you're called the presynaptic neuron. If you are receiving the message, because your dendrites are here, you're called the postsynaptic neuron. But if I'm this neuron and I receive a message, and that message is really exciting, I might want to send that message along. And that's what brings you to, oops, I guess I didn't include another thing for it. That's what brings you to the idea of there being more than one synapse in a row. So we get to draw again. Oops, let me get my pencil. We get to draw again. This time, I want you to draw 3 neurons. So here's 1. Here's 2. And here's number 3. And here I'm going to give them all nuclei because it looks like little eyeballs. Before today's class, or before you started this guided lesson, I bet you never thought you could draw neurons, right? If I can draw them, you totally can too. So here's our neurons. This is maybe a little bit more accurate to real life. We're seeing more neurons in a line. A lot of times it's going to be more than just 3 though. 3 is a pretty short circuit. There's usually a lot more of them. But check this out. In this picture, I have 2 different synapses, right? So I've got synapse number 1, the first place where neurons are meeting with each other. And then I've got synapse number 2, the second place where they're meeting with each other. When we're talking about this neuron relative to synapse number 1, this is my neuron that brings messages to synapse number 1. So this is my presynaptic neuron. This neuron is receiving messages at synapse number 1. If you receive messages, you are a postsynaptic neuron. But here's the kicker, right? Where it gets a little crazy. This neuron receives the messages here at synapse number 1 and says, OMG, I have got to talk to my neighbor about this. This is way too interesting. Suddenly, this neuron is bringing the message to synapse number 2. It's bringing it to a place where it can share its message with somebody else. Relative to synapse number 2, this neuron is a presynaptic neuron because it's before synapse number 2. So it talks to the postsynaptic neuron at synapse number 2, and there's a pretty good chance that this one's going to go talk to somebody else and talk to somebody else, and down the line we go. The big idea with our neurons when we're using these terms presynaptic and postsynaptic, always make sure that you know the location of the synapse, because that's how we figure out if you are in front of it or if you're after it. We need to know what synapse we're talking about. Presynaptic neurons bring a message. Postsynaptic neurons receive a message. And when we put them all in a big line, anybody who lives in the middle is going to be both. It just depends what synapse you're talking about. There is a question on the homework assignment too that helps you to practice this. So presynaptic and postsynaptic, it's all about the location of those synapses. I want to pause for a moment, give us a chance to ask any questions that we have, or to shoot me an emoji. Doesn't have to be a thumbs up. You shoot me any emoji. We're so busy drawing neurons. Let me draw you a penguin. I wish I knew how to draw his arms like he can't believe what he's hearing. That kind of worked. My penguin is gasping. Presynaptic and postsynaptic. Yeah, so Kaylin did a great job summarizing for us. A presynaptic neuron sends the message, and the postsynaptic neuron receives the message. Absolutely. That's a great way to think about it. All right. Well, if we're good, we'll go ahead and move on then. I included this picture just to remind us neurons are excitable cells, right? which means the inside of the cell and the outside of the cell have different charges. I love that this cartoon was just being silly and showing a really excited little neuron that looks a lot like my daughter at about 8:30 at night when I'm trying to put her to bed. But what's cool about it is the fact that notice here on the inside, we have some negatives, and on the outside, we have some positives. Remind me, when a neuron is at its resting membrane potential, what's its charge on the inside? What number do we find in here? What's that number I told you we had to know? Yeah, -70. -70, when a neuron is waiting to get a message, when it's at resting membrane potential, it is at -70 inside. So I love that this little cartoon had a negative on the inside. As much as it was being all cute, it was also being scientifically accurate. An excitable cell is negative on the inside until it starts to do something, and that's going to make it turn positive. The way that the membrane changes is that graph that we started with in our group work activity. So remember, we've got this part of the graph where we are at rest, or what we called resting membrane potential. Where we care about when we're thinking about the way that neurons work is this point number 2 on the graph, where it says a stimulus is applied. In normal person words, or here in anatomy words, this is when neurons hear a message. At this stimulus applied point, this is when the presynaptic neuron is talking to the postsynaptic neuron. This graph, by the way, that we're looking at, this is the postsynaptic neuron's membrane charge. Membrane charge. My neuron who's listening, my postsynaptic membrane, this is what happens to the charge on its membrane as it listens. So we're chilling out here at rest, nobody's talking to me. Suddenly that postsynaptic neuron sends me a message. That message is, I put it here kind of like a lightning bolt, lead to changes in the electricity or the charge of my membrane. If those changes are enough, they're going to push me over that threshold value that we talked about. If I get enough information from the presynaptic neuron, if its messages are enough, I will hit threshold, and that's the point when I, as a postsynaptic neuron, decide I need to go talk to somebody about this. This gossip is too hot for me to pass up. I got to share. So we go up using that process that we talked about called depolarization, all the way up to +30. Now, when I am at the axon terminal, when I'm at the very end of my cell, that I'm the postsynaptic neuron here, when I get to the very end at my axon terminal, I will spit out a message of my own. I'll release my own neurotransmitters. And once I've released those neurotransmitters, I'm going to try to go back down to normal. I'll get a little too excited about being normal, and then I'll get back to resting membrane potential. Every part of a neuron's membrane, from the axon hillock all the way down to the axon terminals, every part of its membrane is going to go through this pattern of going up and then coming back down. That's the way that excitable cells work. They start at one charge, they change that charge completely to do something, and then they go back to normal. So think about this graph as telling you, what do I do to my membrane charge to help my cell talk to its neighbor? That's the goal of this graph. When we're thinking about how we send messages and how we change the charge of the membrane of neurons, now is when we have to go back to some of those Unit 1 ideas when we were talking about our gated channels. Let me ask some of my favorite questions about gated channels. My first question about gated channels. These channels are or are not always open. Gated channels are or are not always open. Yeah, exactly. Gated channels are channels with a gate. There's something that closes them. Gated channels are not always open. When I talk about a chemically gated channel, like we're seeing here, a chemically gated channel, what do I use to open my gate? I promise it's not a trick question. How do I open a chemically gated channel? Exactly. Yep, I've got to use a chemical. I use my chemical key. In the case of a neuron, my chemical key, absolutely, like Timoteo mentioned, is going to be a neurotransmitter. The way that two neurons talk to each other. You and I, we text, right? Or we talk on the phone, very rarely, right? We're always texting. At least I am. I'm introvert. So I would rather text you when I'm ready than have a phone conversation with you. So I'm texting a lot. The way I communicate is I use text messages. The way neurons communicate is they use neurotransmitters. And those neurotransmitters open up chemically gated ion channels. So really, when you think about the way that neurons talk to each other, let me go back to my lovely neuron pictures here. We've got one neuron, give him some dendrites here. We've got one neuron who spits out some neurotransmitters. These are those messages. We've got another neuron who is listening specifically for those messages. Whatever message my presynaptic neuron sends out, I'm going to draw some little chemically gated channels up here. We got spaces on our dendrites where when the presynaptic neuron releases those neurotransmitters, it's going to attach. And when I attach these neurotransmitters to my chemically gated ion channels, I'm going to open up a pathway for ions to go into the cell. There are some questions on the homework assignment that ask you about what actually goes into a neuron when signaling is happening. I want to emphasize with you my chemical keys, my neurotransmitters. Those neurotransmitters are stuck in my chemical gate out here. The neurotransmitters don't actually go inside the cells. This happens too when neurons are talking to muscle cells. Neurotransmitters never actually go inside the cell that hears them. The message attaches outside to the gate. What goes inside are the ions that gate specifically opens and closes for. So keep that in mind. Neurotransmitters always stay on the outside, but they're my key that opens up a chemically gated channel. Depending on the kind of channel that I open, so now we're talking about things like potassium channels or chloride channels. Depending on the kind of channel I open, sometimes that's going to make the charge on my neuron, its membrane is going to go up. It's going to become more positive. Sometimes when I open up a channel, it's going to make the charge on my membrane on the inside go down. Do we happen to remember when I talk about opening up a channel that makes my membrane charge get more positive? What was our abbreviation for it changed my membrane charge and made it more positive? Was that my EPSP or IPSP? when I get more positive. Do we remember which one that was? When I get more positive, membrane becomes more positive. Yeah, there we go. I got a few votes. When my membrane becomes more positive, we said that that was an example of an EPSP. EPSP, that's an excitatory post-synaptic potential. Excuse me. Sometimes what happens to a neuron when a message comes and opens up a channel, when it lets a message inside, what that message does is it's ions that have a charge that make my membrane more positive. I opened up a channel and in rushed, here we'll tie into our group work, in rushed sodium, and my membrane charge got more positive. This little positive change that I see right here is what we call an EPSP, an excitatory postsynaptic potential. My membrane charge got higher because I brought in some sodium. That message unlocked a channel that sodium could sneak inside. I get really excited about that, and it makes me want to talk to my neighbors. Sometimes, though, the message that I receive doesn't make my membrane charge go up. Sometimes the message that I receive makes my membrane charge go down. And that's what we see over here. So sometimes when I get a message, my membrane becomes more negative. Sometimes my membrane becomes more negative. If my membrane charge got more negative because I received a message, I call that an IPSP. An IPSP, that's an inhibitory postsynaptic potential. An inhibitory postsynaptic potential. Remember from our group work that when I open up a channel for chloride ions, chloride is an anion and it wants to come into the cell. If I open up a channel for it, chloride with its negative charge goes into the cell. That would generate an IPSP. That would make my membrane charge go down. If my neurotransmitter, if my message opened up a channel for potassium, potassium rushes out. My positives leave. That also makes an IPSP. My inside gets more negative. So when we're talking about what happens to the charge, because somebody spit out a chemical, we see what kind of gate it opened up. Did it open up a gate that lets positives into the cell? Or did it open up a gate that lets negatives into the cell? Or lets positives leave the cell? That's generating these IPSPs that make us more negative and make us not want to talk to our neighbor. This is going to generate positives and make us more likely to talk to our neighbor. So you have this in your lecture packet. Can you tell me in the chat, which page do you have these pictures on? What page are we at in the lecture packet with these pictures here? Perfect. So I'm seeing 6. This is taking what we just talked about and showing you the specific ions that are involved. So on the picture, here's what I want you to underline, highlight, star for yourself. Over here on my inhibitory side, on my negative side, I want you to circle potassium. My chemically gated potassium channels help me to generate a negative change in my charge. Same deal with chloride that we see up here. Chloride also generates a negative change in charge. And over here, I want you to circle sodium. Sodium is what helps me to generate an excitatory postsynaptic potential, a change in my charge that makes me go closer to threshold. Because here's the big idea with these membrane charges. These are little charges. They change things just a little bit. Maybe we get about 2 or 3 millivolts higher than we were before. Or we get 2 or 3 millivolts lower than we were before. Our goal, if we want to talk to our neighbors, a neuron has to hit threshold for it to be able to talk to its neighbor. So the goal in neurons sending each other messages is, if I want you to talk to your neighbor, I need to open up a channel that makes you get closer to threshold. I need to open up a channel for sodium. So positives can rush in, maybe enough positives to get you up to threshold. Because once you get to threshold, you're committed and you're going to talk to your neighbor. But if I send you messages that take you farther away from threshold, you're going to have to work even harder to get up here to be able to talk to your neighbor. So it's kind of like when someone tells you some juicy gossip and they're like, but you have to pinky promise swear that you're never going to tell anyone. That's like the ultimate IPSP, right? You just pinky promise swore that you weren't going to tell anyone. That's what's going on with an IPSP. Pinky promise swear you're not going to talk to your neighbor. Okay, I'm too far from threshold anyway. I can't talk to my neighbor. I want to pause for a moment. What questions or thoughts do we have about anything we've talked about today? I'll open up the floor. What's one thing you feel okay with right now? What's one thing we talked about today that you're like, hey, I got that? Audrey says she's got the drawings of the neurons down. I like it. Take the little wins, right? We learned how to draw neurons, labeling the parts of a neuron. That's awesome. Okay. Gerardo's feeling good about presynaptic and postsynaptic. I like that. Hey, I'm going to be honest with you. This just kind of like how last chapter was kind of one of those chapters you had to like sit down with and think about in like 5 million different ways, right, to understand it. I know it sucks, but this is another one of those chapters that's like that. So it's totally okay if you're feeling like your mind is blown right now and you don't understand everything yet. It's going to take a lot of review. So I will review this again tomorrow. We'll review it again before the exam. Remember how I always tell you, go stand in front of your mirror and try to explain this to yourself? This EPSP and IPSP stuff, that's a good thing to try to talk your way through, because we got to make sure we understand it. I know we hate those understand questions, right? But we got to understand how it works. Yeah, Audrey says that she talks to her husband about it. I love it. Yep. My husband knows way too much anatomy for a normal person. or that's not fair, because I know way too much anatomy for a normal person too. But I guess I could say he knows way too much anatomy for a normal spouse. We'll put it that way. Okay, well, we'll talk this out some more in group work tomorrow too, because this idea is not going anywhere. IPSPs and EPSPs and those ion channels, they ain't going nowhere. We're going to keep working on them. We're going to skip that for now and come back to it. What I want to do, I'm going to bounce just a moment. We'll come back to some of the things. Is I want to go to this picture here. Can you tell me what page of the packet this picture is on? Okay, I'm here on page 9. Perfect. Okay, so we'll back up. We'll talk more about the summation stuff because this picture is showing me summation. But here's what I want to show you here. We just talked about EPSPs, which are changes in my charge that make me more positive. We also just talked about IPSPs, and that's when membrane charge changes and it gets more negative. The thing with neurons, and you can see it in our picture up here, the thing with neurons is that they're never just hearing one message for the most part. Notice how I've got dendrites all over my neuron right here, right? And notice how you can see a bunch of different axon terminals attached to these dendrites and attached to parts of my neuron here. Neurons are usually listening to more than one neighbor, and they have to use the information from all of their neighbors to figure out if what they're hearing is true or not. So it's kind of like how in good news reporting, right? They have to corroborate their stories. So they need more than one source. That's kind of the way it goes with our neurons too. So let's say that my neuron got one neurotransmitter that opened up a sodium channel, and that made it kind of positive. And the neuron's like, I'm kind of excited about this message. I kind of want to talk to somebody, but I'm not that excited. I'm not to threshold yet. I can't talk to anybody. I'll just keep listening. Well, then somebody else comes along and sends another message that opens up those sodium channels, and I'm even more positive. I'm getting pretty excited. I'm close to threshold up here. But then I read that news story from another news agency that says, actually, this is not true. And so I was excited, but now I opened up a channel for chloride, and my membrane charge got negative. Well, bummer, I'm farther from threshold. I'm not going to talk to anybody. So then I'm sitting here, I'm waiting, looking for more messages. Oh, here comes another message that opened up a sodium. In comes some sodium, and my membrane charge got more positive. I'm getting more excited, but I'm still not to threshold. I'm still not to the point where I know that I should talk to my neighbor. And then I get another message that makes me feel sad. So I open up those potassium channels, and I'm more negative again. But then I have this moment where I get some message that's so exciting. It opens up so many sodium channels that a whole bunch of sodium comes inside, and I hit threshold. Threshold is the magic number. If a neuron gets to threshold, that neuron 100% of the time is going to go all the way up to positive 30. This is like end of the road for a neuron. If I hit negative 55, remember that my voltage gated sodium channels open up. And when they open up, there's none of this back and forth business where we go up and down and up and down. When voltage gated sodium channels open, we're going all the way up. We're going straight up to positive 30. So when neurons listen to more than one message, they're listening for trying to get to threshold. So one message opened up some sodium channels and helped them get a little more positive, and another message made them a little more positive, but then they receive a negative message that makes them more negative. Neurons have to listen to more than one message. If those messages come together and get that neuron to threshold, it's going to talk to its neighbor. If that neuron never gets to threshold, guess what? It never gets to talk to its neighbor. So when you're thinking about the way that neurons work, please keep in mind, threshold is like the ultimate goal. I'm trying to get the threshold so I can talk to my neighbor. If you send me messages that make me more positive, I'm going to get there. If you send me messages that make me more negative, I am not going to get there. This process of listening to a whole bunch of different messages, this is a process that we call summation. Summation. So basically what summation is adding together all the different kinds of messages that we receive to decide if we should be talking to our neighbors. So let's bump back. Now that we've looked at the way that summation looks on a graph, let's bump back and talk about the different kinds of summation that your neurons actually do. So let's go here. What page am I going back to now with this? Excuse me. Okay, looks like we're back on page 7. Awesome. Okay. So we have 3 options of the way that neurons could work here. First option that we see up here at the top is, let's assume that this neuron right here only listens to and only talks to one other neuron. And that one neuron that it talks to only sends messages once. And then it waits a while, and then it sends another message, and it waits a while. I like how we've got the analogy picture over here of a neuron in a rocking chair. When neurons talk to each other and they're making EPSPs, excitatory membrane changes, we're getting more positive. In neurons, we use this word called firing. So if a neuron fires, its membrane charge gets positive. When neurons talk to each other, they're firing. So imagine that you're sitting in a rocking chair, and every time you lean forward, you say the word fire. And then you rock back, and you say fire. Or if we don't want to use the word fire, we could change to talk. So you rock forward and say talk. And then you lean backwards, don't say anything for a while, and you say talk. If a neuron just gets one message, and that message doesn't bring it to threshold, when a neuron doesn't get to threshold, we have these channels in it. We talked about these way back in Unit 1, called leakage channels. Leakage channels, one of their goals is to make sure that you stay around resting membrane potential. Yeah, Kaylin's right. Leakage channels are always open. So I heard this message right here, and it made me kind of excited, but not excited enough to get to threshold. Well, while I'm waiting to hear another message to get excited, my leakage channels are like, yeah, we're going back to normal. We're going back to resting membrane potential. Between my leakage channels and, see if you remember this guy, the sodium-potassium pump, that was my other thing. These two work together to get me to resting membrane potential. So I heard a message, I got excited about it, not excited enough. Over time, I'm going back to resting membrane potential. When we get another message, it'll make me excited again, but if it's not strong enough to hit threshold, I'm going to go back down. If a neuron is not doing summation, if it's not processing more than one message, it probably wouldn't fire nearly as often. It's probably not going to talk to its neighbors very much because it takes a lot of messages to get us to threshold. It's not easy for a neuron to go from -70 to -55. One message by itself is probably not going to get a neuron there. This is when we bring in something like temporal summation. Say instead of going really slow in my rocking chair, forward and backward, say I'm like super excited. Again, I always like to use analogies with my daughter. So this is when we go to pick her up from school and she's like, cheese cubes, cheese cubes, cheese cubes, cheese cubes, cheese cubes, like one track mind 10 million times, saying the same message as many times as possible in as short a period of time as possible. That's temporal summation. Temporal summation. One neuron sends the message repeatedly or really close together in time. So if you think about temporal, that relates to time. With temporal summation, here's an important idea with temporal summation. Temporal summation is one neuron sending lots of messages. Messages, plural. One neuron sending lots of messages. With neurons sending lots of messages, neurons only have the ability to send one kind of message. So what I mean by that is either you are a neuron that's telling your neighbors, you should go talk to somebody else, or you're a type of neuron that always says, don't you dare talk to anybody. There's only two options. So either you are sending a lot of excitatory messages that makes that neuron want to talk to somebody else, or you're a neuron who sends a lot of inhibitory messages. When we're doing temporal summation, you're hearing a lot of the same thing. Either a lot of excited messages or a lot of not excited messages. We're never going to hear a mix. It's always going to be one or the other. Yeah, Kaylin's mentioning a great example when you've got teenagers in a relationship, right? And they're texting each other. It's a whole lot of I love you, I love you, I love you. Absolutely. That's all they say to each other, right? Until they break up. And then it's I hate you, I hate you, I hate you. So excitatory messages, inhibitory messages, we can only get one kind of message when we're doing temporal summation, because one neuron is sending the same message over and over again. When we're doing spatial summation, in spatial summation, more than one neuron is talking to our neuron now. So in spatial summation, we are adding messages that when we're thinking about the space on a neuron's dendrites, more than one of these spaces is getting messages. So spatial summation is when we have more than one neuron sending messages. In the case of this picture, all of these neurons are sending happy messages, but that's not always the case. Spatial summation is not always the same message. With spatial summation, here's the key. In spatial summation, it's more than one neuron talking. Spatial summation, across the space of a neuron, I'm adding together all the messages I get in different places. With spatial summation, some are excitatory and some are inhibitory, or it has the ability to be mixed. There can be more than one type of message. Because remember, in spatial summation, we've got more than one neuron doing the talking. Some of them are excited and want you to talk to your neighbor. Some of them not so excited, telling you to be quiet. Yeah, Kaylin mentioned that spatial summation is kind of like a social butterfly. I like that. Yeah. So we've got the boyfriend-girlfriend thing going on with temporal summation, right? Where they're only talking to each other. And then you've got the social butterfly that's doing spatial summation and talking to everybody, getting everybody's messages. I like that. We will talk more tomorrow about the texting analogy because I'm a little short on time today, but here's what I want to close with today. This is the picture we just looked at right here with spatial summation, how we had several neurons that were all sending excitatory message. All of these were saying, you should really talk to your neighbor. Like, this is exciting news. But in reality, and like that picture that we looked at before, in reality, we've usually got some neurons talking to us that send inhibitory messages and some neurons talking to us that send excitatory messages. So spatial summation, like I mentioned, this has the ability to have both EPSPs, excitatory, positive messages, and IPSPs, inhibitory messages that make our membrane charge more negative. We can only do that with spatial summation when more than one neuron is talking to me. Because remember, each neuron that talks to me, they only ever say one thing. This one can only ever say that we should fire. This one can only ever say that we shouldn't fire. They can't ever change their message. So spatial summation, when you're working on the homework and it talks about seeing a mix of EPSPs and IPSPs, that's what happens in spatial summation. Otherwise, if we're just getting a lot of the same message, if we only get one message, either that's coming from one neuron, and we call it temporal summation, or we've got this confirmation bias going on, right? Spatial summation, where more than one neuron, they're all saying the same thing. Here's the picture we'll land on. In reality, all of our little sketches have looked so clean and so nice, like, oh, there's 3 neurons talking to each other. Yeah, actually, each of your neurons talks to so many different neurons at a time. I know that I have seen somewhere in the literature something like 50 or more neurons that neurons are all talking to. So yeah, I like it. Audrey's like, hey, it's a group meeting. Yeah, so we got the group meeting going on here, where there's all kinds of different messages, all kinds of different neurons that could be talking to us, whether they're telling us to get excited and talk to our neighbor, whether they're telling us to not talk to our neighbor. All of those things come together. We add all of those messages together, and once we've added them together, if we hit threshold, it's kind of like the light switch goes on. We're going to share our message. If we don't hit threshold, that light switch stays off, and we're not going to talk to each other. So we'll talk more in class tomorrow about how a group text, like the GroupMe that Kaylin mentioned, that can be a great example of spatial summation. We'll talk a little bit about how you get a spammer where they just text you all the time. That's a lot like temporal summation. And then we'll talk some more about the way that neurons work and those channels and all that jazz. Any last minute questions about our content from today before I end our recording? I should really like redraw these pictures with penguins. I see all these cute little neurons, but I'm like, you know, they'd be cuter if they were penguins. Edith's good. I suspect most of us are mind blown. Again, it's going to take some processing. All right, well, between now and tomorrow's class at 1 o'clock, try to complete a couple of attempts on part 1 of lesson number 10. Try to at least look at the rest of the guided lesson to get you ready for class tomorrow. That's going to be the stuff that's in part 2 of the guided lesson. So we will wrap up our lesson number 10 tomorrow. I hope that I see most of you then.